Welcome back. Hope you already had made some new connections while the lunch break. May I remind you all to close your cameras and mute your microphones, please? And let's continue to our second session on synthetic biology, moderated by Dr. Vipul Sharma, working at Tampere University Biomaterials Group, Finland. Dr. Sharma, floor is yours.
Thank you. Welcome all and good afternoon. I'm Vipul Sharma and I'm working in bio-inspired functional materials for robotics applications. But today, let's talk about synthetic biology. Synthetic biology is a field of science that involves redesigning organisms for useful purposes by engineering them to have new abilities. Synthetic biology researchers and companies around the world are harnessing the power of nature to solve problems in medicine, manufacturing, and in agriculture. Uh, I was making a quick Google search this morning and I found that synthetic biology is about to disrupt your industry. That was the main line that was there. So it may be a phenomenon without a commonly accepted appellation, but synthetic biology or synbio, what researchers call it, has become a disruptive force that's birthing the bioeconomy. Biology is usually defined as study of living things and life itself, but synthetic biology has turned the science into the manufacturing paradigm of the future. So let's not waste any time, and I'll welcome the first speaker of the day, Professor Nonapa Nonapa. Dr. Nonapa heads the Precision Nanomaterials Group at Tampere University, where he is an associate professor since 2020. From 2017 to 2020, he was an adjunct professor in soft matter microscopy at Aalto University. He was a research fellow in the Department of Applied Physics in Aalto University from 2012 to 17. He has completed his PhD from India's most prestigious institute, ISC Bangalore, and then he was a postdoctoral researcher in University of Uvascula. Uh, he has published more than 80 articles in international journals and has more than 100 conference presentations. His current research interests are biomaterials, nanoparticles, breast cancer, and cryo-TM. And today he'll be talking about pre-models in breast cancer. So welcome, Dr. Nanapa.
Thank you, Vipul. Good afternoon. And I have the big task to keep all you awake after very good lunch. I think that my presentation is more related to the previous session, which was biomedical. And this is very interesting because what I'm going to discuss you today is about breast cancer and specifically about primary tumor and primary solid tumor. And I will ask also see why we need to discuss this topic and do we need something, and specifically I saw that we need and redefine preclinical models. And while redefining preclinical model, I will also show that there is an increasing need of material science, how to combine material science knowledge with the cancer biology and to make some radically new discoveries and overcome the challenges. This has been the work for me. I have a background in organic chemistry, but I have changed my field going to nanoscience and material science, moving increasingly into solving some of the fundamental challenges in cancer biology. And this, I had an opportunity 8 years ago to really participate in the collaboration. And you will see there are more than 20 people involved, multiple institutes, Helsinki University Hospital, and several of these projects initiated through one of the research to business project that I had from Business Finland. Instead of keeping the science, I simplify it, still keeping the scientific meaning intact, and more go in a very general way. So, breast cancer is one of the most leading cancer type in terms of incidence today. Until 2020, lung cancer was the leading cancer, and today about it is the biggest economic burdens to the society, costing about 2 billions of US dollar. That 2% of our GDP. In Finland, we have about 5,000 new breast cancer cases every year, and for this population, it is really important and it's very high. And we have more than 1,000 breast cancer surgery every year in Helsinki hospital alone, and this is increasing. That's because the average age of Finnish women is 45 as of now, compared to India, which is 28. So India is still young, and Finland, the incidence rate of breast cancer is increasing from the age of 40. So that's why it's really important to focus on this topic. However, we often heard today also the clinical trial fails, and the clinical trial fails at the most expensive stage of uh, all the drugs fail at this stage, which that is why we have an overall approval rate. There is also likelihood of approval rate that can be slightly higher, but overall approval rate in cancer drug discovery is less than 4%. And each failed drug cost more than 1.4 billion euros. And in cancer, the one size fits all concept has the least success. And the main reason for failure is the efficacy. That means you have fantastic technical model, works everything perfectly. However, when you go to the real human and a bigger clinical trial, you see that it's not efficient. And other reasons are safety, strategic issues, commercial issues, or operational issues. It is fundamentally challenging compared to many other cancer types, breast cancer, because it's highly heterogeneous and it's complex. It means that two patients can have same symptoms, they have similar type of breast cancer, but they will not respond similarly. And this is due to intertumoral and intratumoral. That means patient to patient and within the patient. And I have a case when we went to discuss with a private clinic that the doctor asked me, I have a patient with the left breast, she has a cancer type. Is responding to hormone therapy. However, right, she has the most aggressive type. And that's exactly what we are interested to understand, how to keep the heterogeneity in the in vitro models. So in overall, 70% of the breast cancer are known as something called luminal type origin. I will explain a little bit later. And 20% is HER2. And these are based on some of the receptors that are overexpressed in particular cancer type. And 10% is triple negative breast cancer, which are very aggressive. They do not express any receptors. And here it's slightly a scientific, but the idea is to really understand what you mean by um, uh, uh, breast triple negative breast cancer. Maybe I can even go here in this. I think I'm loud enough to ask. What we are looking at is a luminal type of cell lines here, and surrounded with this are the basal cell lines, and then you have this blue, which is a basement membrane, and around that is extracellular matrix. We have heard a lot about extracellular matrix today, and I said it's highly heterogeneous. And if you look at the rightmost, there are four types. That's a luminal A, which expresses certain hormone receptors, ER and progesterone. And then luminal B, which is slightly aggressive than luminal A. Then we have human epidermal growth factor. They are much faster growing than the luminal subtype. And then we have triple negative. One of the important challenges to understand the origin, where exactly it originates. If 70% of the cancer comes from luminal, is the luminal cells are the real origin for this. And then bigger challenge happened in Helsinki in our collaborator. They were working for 5 years, thousands of patient sample. They were not able to keep these tissues and solid tumors outside the human body. They undergo transition. They transit into luminal to basal type. Then how to prevent this transition was the biggest challenge. And that's exactly the role of material science comes here. And even more important is that now it will also help us to develop a new preclinical model. The existing preclinical models, they're fantastic for many fundamental studies and give a great clue for drug discovery. However, they are not that complex and are not able to mimic the heterogeneity and complexity, which actually lead to what we call as non-optimal drug identification. And what we do here is that we have elective surgery happening in Helsinki University Central Hospital, and the patient tissue is taken, and then it undergoes some of the pathophysiological process, pathological process, and then we embed it in a three-dimensional hydrogel matrix, and we monitor them. We monitor them by imaging and sequencing before, during the time, and after. So we check whether we are able to keep this type of patient tissues, which is called patient-derived explants. And in this case, explants are fragments of 50 micron size. So from one patient sample, you can make multiple fragments. Therefore, if you are able to keep exactly like in a human body, you are able to test multiple drugs, which you cannot actually do in patients. And what we do is that we also compare this with a variety of other models, such as mouse mammary tissues. We also take healthy breast tissues from reduction mammoplasty and then do all these studies, viability, identity, quantification, the most advanced sequencing. This will allow us to understand the molecular pathway and drug response. So one simple way to understand whether you have a certain type, whether it's a luminal or basal type of cell lines, is using some of the markers, such as they express certain cytokeratins, which are very specific. For example, to luminal, you can have a cytokeratin 8 type of marker, which appears as red if you do immunohistochemistry. Basal will green. If you have a combination of both, a 50% green and red, you will have yellow color. So through this, we can follow some of the changes that actually occurring during your cell culture. And as a first step, here is the most used matrices called matrigel. And this is coming from mouse sarcoma, extracted from this. One of the fundamental challenge in this is really they are undergoing batch to batch variation. They have unknown growth factor, and you can never get the same type of material again. Since there are unknown growth factors and also maybe endotoxins, this result in what we call, especially when we take, here is mouse mammary epithelial cell line, which is means the mouse breast tissue. You take healthy tissue, it's on 0 day, what you see red, it's a luminal type. And after 2 days, you start to see a transition. After 7 days, you still see more than 50% are undergoing transition into basal type. That means we are not able to keep the original cell type in vitro models. What happens if you take human reduction myoplasty? It's a healthy breast tissue, and this is 0 day, and then you have 2 days, almost everything is going towards basal transition, and 7 day everything has transition into basal. That means if you use this kind of cell culture matrix and this model, you are not able to study any drug response. You are not really looking at the current model. So how to prevent this transition, and what actually is the responsible factors for this transition was our question. That's exactly once we solved. I really thought that this is going to be one of the new model. And here, without discussing further, I would like, so it is categorized here that we can make a variety of gels or matrices or culture models. And here we can control one important factor is a combination of mechanical property, which we call stiffness. Stiffness is nothing but the ability to tolerate external deformation or perturbance of any materials. And we can control this by tuning the chemistry and functionality, as we heard from Women's talk this morning. It's a fantastic introduction for this, so I don't have to discuss more. And we can have a category of gels that can keep, for example, luminal identity, and only for, say, patient tissues, but also for mouse tissues. But there are many that works. If you look at the left side, you can see we have a green going towards fully green matrigel, and that is which is changing its identity. Then we have an intermediate matrices. The second row is the patient derived explants, and then we can see that by controlling the mechanical property, we can prevent the transition. But this is not enough. This is a fantastic model. We prevent now, we can keep it 2 weeks outside human body, exactly like it is in the human body, supported by sequencing data. But the next bigger challenge is also to keep the hormone receptor activity. Most of them lose. That is why bio banks are not really useful, because even the biobank stored samples lose their ability to keep the hormone receptor expression. Here, by still controlling the stiffness, we are able to keep this hormone receptor activity. And then it also helps us to redefine how the mouse model actually work, what type of model it is, how human membrane model actually works in different extracellular matrix, providing a fantastic idea that why we really need to go better patient-derived explant cultures. This work has started already in 2015. We had two research to business funding. There was a possible clinical trial, but it has halted because of the COVID, increasing COVID case in one of the private clinics, and that has now diverted into some other direction. But I would like to tell two more slides here that there is no need to worry if you have a primary breast cancer because it is curable up to 80%, provided you do proper screening and have the medication and medication works for you. But 90% of cancer-related deaths often from metastasis. It is a multi-step, highly complex process which is almost impossible to cure, at the same time impossible to develop any model. And exist model, this invasion, extravasation, and colonization in the secondary side. From breast, it's mostly lung, liver, brain, or bone. And how to do this? And the other important factor here is that only in 1 in 100,000 of cells have intrinsic ability to metastasize. So why they undergo then rapid metastasis to other places, and what are the factors? And for this, again, this model doesn't work. We are developing what we heard about lab on chip, but really modular, because we have excellent patient-derived model now we can use to induce metastasis. This is the ongoing work. With this, I would like to thank you, and I'm happy to answer any questions from you.
Invite questions from the audience.
Yeah, thank you so much for this presentation. So, one thing I am interested in all, you are encapsulating the tissue inside the gel. So, how it is different from the organoid or hanging drop culture?
Yes. Organoids are great models, but not for breast cancer because it cannot have that complexity you cannot generate. And this is different because it is the original tissue from the patient. So we know that exactly what is there, both from imaging and sequencing. And this will allow us to follow. For organoids, you don't have any reference. And other fantastic thing that if you develop this kind of model, you can do the drug response in vitro. At the same time, also you select what is the best for patient that can be then given to the patient. This is what we call as patient specific treatment, and it is called by different name, personalized or precision, but it's more about patient specific. At the in the longer term, say from several years from now, it is possible that you can have several patient specific treatment. Then you can group them together depending on their response, or for example resistance for certain treatment. Organoids don't do this job for breast cancer. They are nearly not so useful. They are useful to get the ideas and find some lead candidate, but not for really do this precision treatment. This kind of models will help you to do parallel thing. But again, this is only for solid tumor, not for liquid biopsy, not for metastasis, and specifically for luminal. That's why I wanted to make sure that I'm talking about primary luminal type tumor.
Very interesting talk. Just one simple question. As you mentioned that in matrigel, you see the transition from basal to luminal, as well as in other AC-derived matrices, you might also see the same.
Yes.
But why do nanocellulose hold the features phenotype and not does not undergo transition from one function? What is the reason behind that?
Unfortunately, nanocellulose don't hold. And what I have not shown nanocellulose, these are different biomaterial based. Nanocellulose is not really good, unfortunately, for this type. And it is good for many other. We have seen, for example, some other work ongoing on TNBC type of cell lines, and whereby changing certain properties, surface property we can do, but they are not good. I have shown the example of Grodex, for example. It doesn't work for, it may work for some cell line, but not patient tissues, not even for mouse memory. 2 days transition. Thank you. The reasons are multiple. There are stress-related activation of something. If you have high stiffness, that is necessary. If the stiffness is low, maybe there is no such activation. And secondly, it also has to do with some biochemical cues, not only mechanical. It's a combination of biochemical and this. And this is a big debate in the literature. Even today, there is a controversy whether all cancers, breast cancer, originate from the luminal cell line or how they transition, because luminals, of course, can transit, or is it from basal, because TNBCs are basal type cells. They do not express any hormone receptor. They are triple negative. So whether it's actually originating, because if you take the cancer type, if you are able to map the pathophysiology, you can see that many of them are heterogeneous. You have certain percentage of basal also, depending on the stage at which the diagnosis is done.
Our second speaker of this session is remote, Professor Pawan Kedhar. Professor Pawan Kedhar heads Synthetic Biology Group at the School of Biotechnology, Jawaharlal Nehru University, New Delhi. He's also the co-founder of Foresight Biotech Private Limited with a focus on drug discovery, and Clear Meat Private Limited with a focus on lab-grown meat. In 2007, Professor Dhar established Systems and Synthetic Biology Journal with Professor Ron Weiss, MIT, and ran it for 9 years. Some of his key contributions are Clear X9, which is a slaughter-free, effective, and affordable alternative to fetal bovine serum. Flagship, which is a novel biotherapeutics platform originating from the dark matter of genome, and Synthetic Biology Governance Framework for the Indian government. In the past, Professor Dhar has established research groups in India, Singapore, and Japan, and published in the area of human genetics, systems biology, and synthetic biology, covering computational, experimental, and policy areas. Today, he'll talk about the dark genome. So let's welcome Professor Dhar.
Well, you see, when you talk of synthetic biology, where does it come from and where do we fit into this? I need to take a few steps back and trace the origin of the whole idea and then come back to our work very quickly. So you see, when you do reductionism of the organisms or the systems, what you get sequentially is first gross anatomy, histology, then cell biology, molecular biology, biochemistry, and so on. Now, when you get a lot of data as a part of the reductionist process, you got to integrate it back. You got to handle the data. And the integration of data gave rise to systems biology. And then 1.5 decades back, engineers at MIT and elsewhere in the US, biologists and engineers together, they said, why don't we construct the system instead of integrating and instead of doing the reductionist process, two different approaches to do science. Well, this gave rise to synthetic biology, which is also known by the name of biological engineering. The central theme in this meeting and the follow-up conferences was that you create standard parts, and from standard parts, you create standard devices and standard circuits. And you boot up a system, you construct it ground up, and you basically engineer biological systems. So, though we have a term like genetic engineering historically being used, but frankly, we never engineered genetics. This is the first time that we are deliberately trying to engineer biological systems. So the focus was on building standard parts. However, we have taken a slightly different approach to this, which is also matching the whole idea of construction. But we said, instead of using existing parts, that means existing genes, existing RNA, existing proteins, instead of using them, why don't we create new parts? Why don't we create new genes? Then how do you do that? Well, if you see, functionally speaking, there are 3 kinds of sequences in the genome. One is coding for protein, the other codes for RNA, and there is another segment that does not seem to code for anything based on the best annotation efforts. And so this fraction of sequences that is non-expressing, we focus on that. because the RNA coding and the protein coding sequences have received a lot of attention for several decades. So the point was that why are these sequences there? And what will happen if you delete them? What will happen if you synthetically make new genes from sequences that have never been expressed by nature? They are kind of hand-me-downs from the earlier organisms, but do they make sense in biology of an organism if they are not expressing at all? Or is this a kind of a gold mine that has not been tapped? So these questions gave rise to the Dark Genome Project. So dark genome, the way we define it is the sequences. There are three types of sequences in dark genome. One is the sequences that are never expressed in evolution. So you have absolutely non-expressing fraction. And the second type of sequences are those that do only RNA expression. They don't go to the protein level. And the third type of sequences are what we call is virtually dark. That means they were expressing ancient times, let's say pseudogenes, and something happened, mutations, and they're not expressing at all. So the question was, can we make brand new genes from these three types of sequences? So this question then triggered a project when I was, back then in from 2006, we started this work. I was at RIKEN Genome Science Center in Yokohama, and the synthetic biology work was funded intramurally by RIKEN. And so we went on a slightly risky path because there was no publication at that time that would establish that this approach would be successful. So we had a lot of trouble in convincing people to do this project. But because we had strong intramural funding from RIKEN, it was slightly easy to go ahead with the idea. So the proof of the concept came up when we were able to make functional proteins from the intergenic sequence of E. coli. One would easily discard these sequences in the sense of experimental biology because we don't normally consider intergenic sequences when we do the functional genomics. But this was the first report published back then in 2009 that established that the intergenic sequences can also be used to make functional proteins. And then we found that one of the proteins is actually killing the cells. And because it was killing the cells, we asked if we switch off the protein expression, will it restore the cell growth? And that's exactly what happened. I'm sorry, I don't have the slides and results to show you that. So by switching off the gene expression, which is coming from the intergenic sequence of E. coli, we were able to restore the cell growth. And then the next obvious question was, what are these guys doing in the cell in terms of the structure? How are they looking like? Well, we had to stay with the computational prediction, even though we had 42 NMR machines, but all of them are busy with the Protein 3000 Grand Challenge project in Japan. So anyway, we went ahead with the computational predictions and published this paper. And following that, we were curious to know what is the boundary condition of this approach. Will it work all the time? Now that you're looking at not 1.5% of, let's say, human genome is protein coding. What we are talking about is 98.5% of human genome. Can we synthetically convert 98.5% into protein coding genes? It's a huge space. It's a large space. So we had to do bioinformatics because probably first time we were fortunate to get the results, but maybe next time we will not be fortunate. So we went ahead with the bioinformatics project, and then we selected certain sequences. And one of the surprises was that the Drosophila intergenic sequence, which looked very different from antimicrobial peptides in terms of sequences, in terms of the structure, it was nearly similar. So you have two sequences. One is coming from the marketable antimicrobial peptide, and the other one is coming from Drosophila intergenic sequence. Both of them are looking different when it comes to sequence, but structure nearly similar. So we then chemically synthesize the peptide, and it shows a very strong antimicrobial property. Then we generated another proof of concept by looking at yeast sequences, yeast intergenic sequences, and we made some anti-malarial peptides, and we found that in presence of these peptides, 60 to 70% of malarial parasites are unable to enter RBCs. So then we generated another experimental evidence. So we asked, how about making peptides from tRNA? Remember, tRNA has a historical role in ferrying the amino acids to the ribosomal interface. So it's kind of doing an outreach program for billions of years. So we said, if this tRNA had an opportunity to express its sequence in terms of translating its sequence, what kind of peptide would be made? So after a lot of bioinformatics filters and so on, we were finally able to synthetically make certain tRNA peptides, tRNA encoded peptides that we call as trep. And we found that they are showing very strong anti-leishmania property. The IC50 value is 40 nanomolar range. And the best part of the story was that if you increase the concentration of these tRNA peptides 10 times, human cells are not affected. Only the parasite gets affected, which is anyway very strong inhibitory effect. And then recently, we also generated another proof of the concept by transforming certain intergenic sequence of E. coli and generating some anti-Alzheimer peptides. And we found that we were able to suppress the base expression by 95%. And the peptides are coming from the intergenic sequence. They were never expressed in nature. So this has now generated a lot of excitement and spin off a company called Foresight Biotech Private Limited. Well, there are a lot of other things going on. We are in the process of making molecules, functional molecules from reverse coding sequences, which means that when you reverse the frame, that is -1, can you get, that means the protein coding genes, when you reverse the frame -1, can you get functional molecules? So we are in the process of validating some of very interesting molecules. Likewise, we're working on antisense strand, making genes from antisense strand and trying to see the function. So the entire space is very, very large because we're talking of millions of such possibilities, not just thousands. And we are using of late some AI approach to zoom into the molecule of interest that we can, well, of course, we cannot do thousands of experiments, so we need to be very careful about what we choose. Now this has been a pretty encouraging story so far. Very recently, we also taken up this challenge of producing the slaughter-free meat and spinned off another company. And as a result of that, one of the product that has just been that has just undergone the soft launch and will be commercially launching it by the end of this year is called ClearX9, and this is a replacement for fetal bovine serum. So as you know, unfortunately, and there's no other way, we need to sacrifice, we need to slaughter pregnant cows and extract the fetal bovine serum for the cell culture. So we asked, can we do something about it? And it took us 3 years to find the right combination of molecules from natural resources, slaughter-free, and we'll be happy to demo it whenever there's an opportunity. So these are some of the areas we are working. In future, we are looking at the possibility of other molecules. There are many interesting topics in synthetic biology that have social applications. So if there are any interest in working together, we'll be delighted to do that. So thank you very much for your kind attention. I know we are very close to the coffee break, and apologies for the inconvenience. Thank you very much.
Thank you, Professor Dhar, for improvising your talk. It was really impressive. and thanks for letting us know about your works. We'll take some questions now.
Yeah, thank you very much. That was really an interesting talk. I have a very simple question. You just said that you are designing this synthetic serum. Uh, but to my knowledge, it's really a very complex constituent and very important constituent of all cell culture media, everything where we do animal cell culturing, everything. So will you be able to, really replace that thing?
Yes, I think this is a great point because having been in the cell culture domain for the last 3 decades, I can tell you it's not easy to replace fetal bovine serum. It's very complex, very challenging. People have tried different combinations and failed. It has not made a commercial dent so far. We also failed. We also faced a lot of failures, and we almost gave up. But something worked one day, and we then distributed this ClearX9 to several labs in and around Delhi. So far, we have been able to get encouraging results. And also, we were encouraged by the fact that even the people who are working on stem cells or nerve cells, they are also able to grow these cells with our medium without fetal bovine serum. So far, there has been motivating feedback, but I'm sure we'll fail somewhere because more and more people need to test this. And if we fail, and I'm sure we'll fail somewhere, some system it may not work, but that'll be a good thing because we're waiting for that, because that will tell us what is happening at the molecular level. And we've done all kinds of characterization, next-gen sequencing, and all of that to see whether it is increasing the cell numbers but making the cell sick in the process. Is that what is happening? We have been able to also cross that barrier, but yeah, we can know only when people test it more and more and see. Yeah.
Yeah, that's really encouraging thing because I remember there's also a Finnish company who is trying to design synthesize egg yolk and synthetic egg, something like that. So yeah, that will be really a pioneer work in this field. Yeah, thank you.
Thank you very much.
And I hope all of you now know what the synthetic biology is. Dr. Anna Haponen, formerly Kakonen, a senior scientist in Luke, is a plant physiologist who's an expert on plant cell wall and lignin biosynthesis. Dr. Kakonen has had an academia research fellowship from 2012 to 2016 and collaborates with several research groups worldwide. She has supervised 5 postdoc fellows, 5 graduate students, and several undergraduate and international visiting PhD students. Dr. Kakonen has a long experience in the studies of enzymes and the source of reactive oxygen species related to lignin formation, and also in various techniques for cell wall analysis. In Luke, she is currently involved in a project that investigates plant exovesicles. She has also participated in project of nutrient uptake of forage grasses, use of cereal hulls in the paper making, and use of hop biomass for applications. Today, she'll be talking about lignin biosynthesis study in Norway spruce. Please welcome Dr. Anna.
Thank you for the invitation for this interesting seminar. Professor Penttila gave us a very nice talk in the morning about biotechnology using microbes. Plant cells can be utilized similarly as hosts for production of proteins and metabolites. Okay, today I will talk about my own field of research, that is lignin biosynthesis studies in Norway spruce. I give you an introduction about lignin, then tell something about our studies, and then briefly discuss how this cell culture we are utilizing could be used for production of certain chemicals. So just a brief introduction to remind you all. So the most is this visible? So the outermost layer in the plant surrounding the living protoplast is the plant cell wall. And that's the difference between plant cells and animal cells. All plant cells have a primary cell wall that is composed of cellulose and hemicellulose and pectin. And then there exists water because all the enzymatic reactions occur in the aqueous surroundings. Primary cell wall allows cells to elongate, and as said, it's present in all plant cells. And here is a schematic picture how the cell wall components are organized. Cellulose microfibrils, these ones. Then there is hemicellulose fibers over there, and pectin matrix is between. Then secondary cell wall develops to certain cell types only. It develops after cell elongation has ceased and cell has attained its final shape. Again, cellulose is a main polysaccharide there. And it develops only in water-conducting cells like tracheids and vessels that conduct water, like you see there are the pipes over here. And it develops to dead supportive cells like stone cells and fibers. Fully differentiated cell is composed only of cell wall. A new component is coming to this kind of secondary thickened cell walls, and that's lignin. It's a phenolic polymer. You see it's kind of a net, and it is deposited between cell wall polysaccharides. This excludes water from the cell wall. Lignin gives strength to the cell wall. It gives water impermeability, and it's a good mechanical barrier against pathogens. And why do people study lignin? It's the second most abundant terrestrial biopolymer after cellulose. Basically, about 30% of the organic carbon in the biosphere is in lignin. So it's a very good sink for carbon. And when the plants colonized Earth, then lignin developed and allowed plants to grow tall. And this is just to remind you that lignin is present in all higher plants. It's not only in woody plants. The upper picture shows an example of a grass. The red stain now tells where the lignin is. In grasses, lignin is present in water-conducting cells, and this red ring around the stem, that's giving very good support for the stem. The next one is Arabidopsis, an example of a herbal plant. There you see also water-conducting cells stained red and also the supportive cells red. And the lowermost one is Norway spruce, and the red color stains that all the secondary wood is lignified. And here is an example again, this Norway spruce. So the composition of the secondary cell wall in conifers like Norway spruce, the most of it is cellulose, 40%, lignin, about 27%, then various hemicelluloses, about 1/3, and then extractives. And depending on the species, the lignin concentration in wood varies from 20 to 32% of the dry weight. And lignin is important to man because in pulping you have to remove lignin to make good quality paper. It's important in biofuels production because lignin hinders the use of cell wall polysaccharides and for saccharification and fermentation to ethanol. In milk and beef production, lignin impairs digestion of cell wall polysaccharides of forage in the rumen of ruminants. So that's why you have to harvest the forage grasses at a certain time when they are not too much lignified. It has advantages in burning because it has a high energy content. And nowadays there are lots of applications under research and also already in market. So for example, lignin can be utilized in panel board adhesives, insulation materials, batteries, antioxidants. Vanillin, the aromatic compound, originates from lignin. You can use lignin in composites, carbon fibers, so there are lots of research on this. Norway spruce is one of the most important forest trees in Finland, and it is utilized for both timber and for pulp production. And for example, in 2014, about 73 million cubic meters of wood was used for wood products, and they contain about 19 million cubic meters of lignin. And in the following year, about 30 million cubic meters of wood was utilized for pulp production. meaning that 8 million cubic meters of lignin was removed. So there's lots of lignin. And how lignin can be studied? Of course, you can study it in native plants, but there are also models. And plant tissue cultures are an excellent model to study lignin biosynthesis. In the upper picture, you see an example that is Zinnia elegans cell culture. Basically, you take a leaf of a zinnia and homogenize it mildly so that the cells are separating from each other. And then you put them to the growth medium with certain growth regulators. And in a few days, you can see that the cells have differentiated, and then they have started to differentiate again. And now they have differentiated to tracheary elements, that is wood cells. You can see the cell wall thickenings on the right-hand side figure. Then there are tissue cultures that produce extracellular lignin. This is our research material. On the left side, that is the time 0 when the cells have been put to the liquid condition. And the following one is after some days, 5 days in this case. The culture medium is cloudy. It's not contamination, it's extracellular lignin. So this can be utilized for research purposes. And what are the advantages? They are controlled systems. They are not dependent on the season. Like when you study native trees, you have to go in summer to forest and harvest the tree. But these can be studied all year round. It's easy to isolate the enzymes from the culture medium and study them. It's possible to add inhibitors or substrates of the putative enzymes into the cultures and follow what happens. If you want to study lignin, if you work with the extracellular lignin forming cell culture, you can easily isolate the lignin. For example, we remove the cells by filtration, and then we can easily pellet the extracellular lignin by centrifugation. And then the structure of lignin can be studied in its native form. This is an advantage because when you extract lignin from the trees, the structure gets modified. And in case your cell culture is transformable, it is an efficient way to test the function of genes of interest because you can transform your culture and see whether the lignin formation is affected, for example. So this is the cell culture we have been utilizing as a model for years. It is maintained as a callus culture on solid medium, the left figure. And for experiments, the cells are transferred to liquid conditions. And why we think this is a useful model? Because the extracellular lignin structure is intermediate in linkage structure to that of mildwood lignin. Mildwood lignin is the lignin isolated from the tree. So the linkage structure is shown here in the table. So basically, when you compare the linkage structures, it's intermediate to that of compared to mildwood lignin and artificially produced dehydrogenation polymer. The lignin has been further characterized. So it was found that this extracellular lignin contains some bound carbohydrates. And that's why it was a useful model to study LCCs, lignin carbohydrate linkages. This was done in collaboration with Martin Lavoko, KTH, Sweden. And they found out that benzyl ether type linkages exist between the lignin and the carbohydrates in this lignin. We have studied how lignin polymerization occurs. The left hand shows the precursors, the main precursors, parakumaryl alcohol, coniferyl alcohol, and synapyl alcohol. Then you have an oxidative enzyme, either peroxidase or lactase, that activates these phenolic compounds to radicals. And then these radicals meet each other and make first dimers, trimers, tetramers, etc., and form this lignin polymer eventually. The question has been, that has been studied a long time, which oxidative enzyme is it that activates the monolignol for a radical? Is it a lactase enzyme, oxidase enzyme that utilizes oxygen, molecular oxygen as an oxidant, or whether it's a peroxidase enzyme utilizing hydrogen peroxide? Because both of the enzymes are able to make similar types of radicals, and that can then make lignin polymer. And how we approach this? We added a hydrogen peroxide scavenger, potassium iodide, to the culture medium and followed what happens. Basically, we modify the redox state by removing the hydrogen peroxide from the medium. The left figure shows the water-treated cultures. So they have formed extracellular lignin in this 5-day culturing period. The right figure shows the iodide-treated cultures. There's no extracellular lignin. We can quantitate this. So basically, the blue bar shows the extracellular lignin that has been formed in control cultures that got a small aliquot of water. The lighter blue bars show that with iodide addition, there's no there is slightly some formation of something, but basically the culture medium stays clear. And we can cultivate the cells up to 4 weeks, and they don't produce extracellular lignin. The purple bar shows the potassium chloride that was used as a control to show that it's not a potassium effect, it's the iodide effect. So in this cell culture, hydrogen peroxide is needed for lignin formation. So in this culture, peroxidases are needed for lignin formation. It might be that it's a species-dependent situation because in certain species, the lactases have the role. But in our cell culture, it's the peroxidases. So we have done large-scale phenolic profiling, and this is the experimental setup. At time point 0, the cells were transferred to liquid condition. And then this big aliquot of liquid was aliquoted to 2 pools. So there was lignin forming conditions, the upper row of flasks that got a small aliquot of water, the corresponding volume that the lower row of bottles got iodide. And then we cultivated these. There is time point every 5 days the collection was made up to 20 days. And in this 20 days time, the water-treated cultures, they got extracellular lignin, but the iodide-treated flasks didn't get it. But then we took the iodide away and washed the cells and put them back to the normal medium, and then lignin came gradually back. So we wanted to study how the phenolics behaved, the small molecular weight phenolics in these culture conditions. So that's why we took the medium, removed the lignin by centrifugation when there was some, and then analyzed the medium contents by LCMS and GCMS for small molecular weight metabolites. And basically, LCMS, liquid chromatography MS, showed that there are small molecular weight compounds inside the cells, inside the protoplasts, and in the culture medium. And what we found out in the cells, we found glycosylated dye and oligolignals. There were lots of different types of flavonoids. There were proanthocyanidins. In the culture medium, that represents the apoplast, basically the fluid that permeates the cell walls, there were dye and oligolignals and glycosylated dilignals. Our approach allowed us to follow how each compound group were kind of accumulating. And basically, we can conclude from this work that modifying the redox state in the culture medium by adding iodide there guide which kind of compounds are formed in cells and in the culture medium. And basically, this can be utilized. Basically, we could produce different kinds of compounds by having this cell culture, for example, by adding iodide there to stop the lignin formation. But in case we want to study the lignin for high-value applications, that could be produced by using this cell culture. And I can conclude that the cell culture is a very useful model for studies related to lignin formation. And we had a visiting scientist, Dr. Anirban Bar from India, visiting Luke in 2019. With him, we did trials to transform the cultured spruce cells, because that is not possible yet. And he did some wet lab work and also transcriptomic data analysis. And we have the manuscript under preparation at the moment. So there are possibilities for collaboration in case you are interested in this kind of work. And I want to acknowledge the people who have been involved. And phenolic profiling work, Teresa Laitinen was the student working on that. And both Buerian was the important collaborator in University of Ghent in that work.
Thank you, Dr. Anna, for really nice elaborative presentation. I have been studying these kind of materials from long time. I still did not able to understand how this cell wall is bonding with the how lignin is bonded with the plant cell wall. Either it is binding with the cellulose or is binding with the hemicellulose. What is the connection where it get deposited?
Well, at least the LCCs, in our case, it was the xylan where the linkages were, but I know that also pectin can make LCCs with lignin. But I think in cell wall, it usually stays in the cell wall because there's no space because the cell wall is there. So there's no liquid to take it away. In our case, because the cells are in the liquid conditions all the time, so for certain reason I have to say that in our case, the cells are not differentiating to tracheids. Only 3% of the cells are differentiating to tracheids, that is wood cells, with a secondary cell wall. Majority of the cells remain as non-differentiated with only primary cell wall. And that might be the reason that the lignin is kind of slot off to the culture medium. But in plants, of course, it can't slot off because the plants are in air. Basically, there's no water to, this is a special case that we have utilized. It's not normally like that.
Yes, we have a question.
Thank you very much for very interesting extracellular lignin. I'm wondering whether you could comment on the structural molecular level uniformity, because one of the bigger problem in lignin, still understanding this millwood lignin, is understanding the structure because so heterogeneous. Nobody really knows the exact structure of lignin. Whether your cell culture will help to solve some of these challenges.
Well, there was this comparison has been done by 2D NMR. This shows the linkage structure, so you can see that also in this, it's the middle row, there's extracellular lignin, so there are different types of linkages. The beta 4 is always the most abundant, independent of the lignin source, but it has similar types of linkages. In our case, our extracellular lignin also has these carbohydrates, and there are also proteins bound to it. But it's not uniform. There are different types of linkages, but basically we have not modified this lignin, so you can kind of trust that these linkages were there already when the cells formed, or it was formed in the medium.
Are there any more questions? If not, yes.
Maybe you said it or not, but I'm wondering how much can you now make conclusions, and let's say time-wise conclusions also, that how is the synthesis going on? You have a transcriptional profiling, I guess, and also in a way that you said that you can measure also some enzymes in extracellular enzymes. So in a way that you could trace in a way the lignin synthesis, and then also towards the complex lignin formation. So would you think that you would be able to do that? And also an additional question, that how much are there spontaneous reactions, which are not enzymatic reactions, which are occurring in the liquid, which could also then be occurring when lignin is formed in the tree?
Well, I start with the last question. Okay, the idea of the enzyme's role in lignin polymerization is that the enzymes make the radicals. And after that, you don't need an enzyme. It's just it is a random coupling that the dimer meets another dimer, radical meets another radical. So that is thought to be, but of course there is, in case of lignin formation in this cell culture, there is a certain level of hydrogen peroxide present when we kind of don't use the iodide. So of course reactive oxygen species can cause reactions. So there might be some non-enzymatic reactions. Okay, we have studied enzymes. They were just examples, these oxidative enzymes. I personally have been interested in apoplastic reactive oxygen species formation. So I have studied those enzymes. But sorry, now I forgot your first question.
Yeah, I'm just the interest in a way that how much on this system that you have, you can draw conclusions how the real lignin biosynthesis trees is occurring, and also that maybe I can phrase it differently, that how much of these radical reactions actually would drive so that you would have certain structures.
Well, when we did the transcriptomic analysis, basically it is the same because the monolignal biosynthesis genes, they are encoded by gene families. And in trees, there is a certain member of the gene family that is responsible for certain step. So in this tissue culture, basically it was the same gene family member that is kind of induced when this lignin is forming. So that is also good for because of course this is model. So some things are happening differently than in the cell wall of native plants. But at least the fact that the same family member was expressed in the situation of lignin formation is kind of making this a better model. But of course you are correct that certain things like reactive oxygen species in the culture medium can cause something that is not happening in the real cell wall.